Okay, so this is fluorescence labeling. The cool thing about fluorescence labeling of anything, typically you do this on antibodies, so you don't have to try to incorporate a fluorescence label into a protein itself, and you can then just attach your fluorescence labeling onto a target section, is it enables the proteins to actually be visualized by fluorescence microscopy, excuse me. God. And this is going to reveal the location that they tend to conjugate into at a cellular location for that protein. So, example, your mitochondria might light up with fluorescence if they tend to be in the mitochondria. This also helps to provide a clue to the protein function, because where it is going to limit what it's exposed to, right? The fluorescent proteins can tag cellular proteins to follow their movement in the cell, so you can actually see how they migrate through different processes, okay? And so you could actually look at cells when they're dividing, when they're doing different things. When you just recently put glucose in, hey, I have a bunch of these now popping up and fluorescing, okay? Which is really awesome. The antibodies labeled with clusters of electron-dense metal can be used in immunoelectron microscopy. This right here is actually a fluorescently labeled monoclonal antibodies, what we're looking at right here. And you can sort of see how this works out. Now keep in mind that oftentimes this is not what you would see directly with your eyes, okay? This is usually a non-visible light that you would see, or light that you couldn't actually differentiate. And then you have a computer program that's highlighting sections, okay? You normally would not be able to see that sort of gradient, really fine, lots of different colors with your eyes in a microscope, okay? That's just not happening. Okay, so the nuclear localization of a steroid receptor, this can be made visible by attachment of a yellow variant of GFP. This is actually then seeing it happen as your receptor is doing its function. So we have 0 minutes, 10 minutes in, and then 60 minutes in. And you can see how the receptor is actually getting flooded and concentrating that. Okay, this is actually the immunoelectron microscopy. Again, we have very, very fine spatial resolution in this case. Notice how you can see basically almost individual particles in there, if you look really closely. Whereas this, you can just see sort of a flooding of light in the region, right? You can see like density variance a little bit, but not much more, okay? So mass spectrometry, everybody remembers MS, mass spectrometry from organic chemistry, hopefully. If you don't, that's okay, just talk to me. This allows highly precise and sensitive detection of your mass of your analyte. Mass spectrometers convert the analyte into gas phase ions and apply electrostatic potentials. This is going to give you your mass to charge ratio, your MZ ratio. Everybody remember that, okay? Okay, being divided by the charge. This is going to consist of 3 components. You have an ion source, a mass analyzer, and a detector. The mass analyzer is that magnetic field that separated your different MZs. And then the detector tells you how much you got. Matrix-assisted laser desorption or ionization, which is denoted as MALDI, that's the abbreviation, is often combined with time of flight. Time of flight meaning you measure how long it takes to get there. And so you can like have GCMS. You can have different variants. You can actually have GC, GC, LC, MS, MS, MS. Okay. You literally just plug each thing in. Gas chromatography to gas chromatography to high precision, and then to an MS, to another MS, to another MS, so you can resolve very fine differences in MZ. Okay. Yeah. Anyways, so wonderful. MALDI, the analyte is going to be evaporated in the presence of a matrix that can absorb the light at specific wavelengths. An electrospray ionization, ESI, a solution of the analyte will then be passed through a electrically charged nozzle, and this will give it its charge. The mass analyzer is going to be where the analyte ions are distinguished on the basis of their mass to charge ratios. That's that magnetic field that's going to select for differences in mass to charge ratio. Depending on the size and the amount of magnetic field you have will give you how much separations you can get, how resolved you can get that. The time of flight mass analyzer, the ions are going to be accelerated through an elongated chamber under a fixed electrostatic potential. Notice this is very different than the one we had talked about earlier, the normal mass analyzer. Okay? Instead of hitting the sidewalls, you're actually looking at how long it takes you to get from one side to the other. Okay? And so the mass of each ion is going to be determined in a time of flight TOF mass analyzer by measuring the time required for each ion to pass through the chamber. The reason you would do these different methods is the mass analyzer actually takes a lot of sample, okay? And so you actually have to break apart and use a lot of sample for this. And you need a pretty decent concentration, right? So for traditional chemistry, that works really well because we can just make more, right? And everything that you're not selecting for at that given moment is wasted, right? It's going to go off to the vacuum, gone, done for, okay? A time of flight, on the other hand, you get stuff that hits first, middle, last. You don't really waste it, right? Now, let me ask you a question. Are you getting these samples back? No. No, you're breaking them apart. They're done for, right? So whatever sample you put in, this is called a destructive technique, okay? Meaning that you're almost certainly not going to get a sample that you can use back, okay? Whatever you put in, it's gone. And for a biological sample, that can be very difficult because if you try to grow a new one, you're not going to get the exact same, even if you clone, right? Even clones are not the same, okay? Okay, so what we have here is we have a sample coming in. Here's our detector, by the way. This right here is our source. So we put in a protein sample here. This is going to be ionized by this beam here that's coming in. The laser beam is actually going to hit our matrix that has the protein embedded within it. Okay? And then what's going to happen is once this is formed into an ion, it's going to have a charge, right? And so it'll begin to be attracted by this right here, which is going to be the head of our ion source. Okay? This is where we have the first electric field. Okay? And so it attracts, in this case, this is negative rings. It's going to attract our positive ions. There's a hole in the middle, so they're going to get catapulted through. Okay? And this accelerates them. They're then going to have a flight tube. This needs to be evacuated, meaning not a lot of atmosphere in it. Otherwise, they're going to get slowed by the pressure, right? So you need to be consistent, low pressure. Okay? Okay. Now, any given amount of acceleration, what happens with our mass? If we have the same amount of acceleration pulling them in any other direction, then down. Down, they'll pull equally, right? But any other direction than down is going to pull differently, isn't it? Okay, so for a given amount of force, if you have a lighter object, it's going to be accelerated to a faster speed, right? If you have a medium object, medium speed. Larger object, more weight, slower, right? Okay. And so they'll all have different speeds. You can then measure the amount of time that takes them to get to the detector, and this gives you the time of flight. Your lighter ions will come out first, your heavier ones come out later, right? You will need some method of standardizing this, by the way, and so you oftentimes will have, I would prefer an internal standard here, and normally they use internal standards, but you can standardize it once and hope. Just a bad idea. This would have a recorder, by the way, that's going to tell you how long it takes for that flight. And that recorder has to be linked to your pulsing the laser, right, in some way. Typically, this is through this right here, which is the beam splitter, okay? And so when you pulse the beam through using the beam splitter, when it's off angle, it doesn't allow the beam through. When it's on angle, it allows the beam through, okay? And then this will give a little bit of the beam, hits a time recorder, starts the time, and that tells you how long you're taking until this is hitting over here. Okay? Anybody have any questions about that? All right. This right here is a multi-TOF mass spectrometer, which is what we just talked about. You can see here we have insulin and beta-lactoglobulin, and you can see there's actually a decent resolution of these. Okay? These are the peaks that you're actually seeing. So here we're actually getting an additional H+, right? And so that's that extra bit of mass, and it has a 1+ charge. Everybody recognize how to read these peaks again? Okay. The L here is the lactoglobulin. I here is the insulin. And so, for example, this one has taken on additional hydrogen and has a 1+ charge, right? Okay. And that gives that mass over charge ratio right there, right? Its mass essentially being +1, right? Okay. Note that you do very oftentimes get the additional plus hydrogens coming on these things. They're fairly large molecules. And so you do have to watch out for these small peaks with the additional hydrogens. Very common. Okay? But your primary peaks are typically just going to be 1 hydrogen for most things. The Edman degradation method has mostly been replaced by mass spectrometry, as we just discussed. But you can still use the Edman degradation method, and do not think that these past tests are done for. Okay? When you go to calibrate a new method or go to calibrate a machine you haven't calibrated before, oftentimes they'll be required to run one of these old tests. Okay? Because they're very reliable. Step 1 is you're going to take phenyl isothiocyanate, PTH. This is going to react with the N-terminal amino acid of a polypeptide that will form the PTH amino acid derivative. Step 2 is going to be the cleavage. This is going to release the PTH amino acid without cleaving the remaining protein. Basically, you attach this, cleave off the amino acid. Okay? Step 3 is going to be degradation, and this is going to be subsequently repeated over and over and over. And so the chromatography here can then be used to identify your specific amino acids you cleave each time. Notice this can take a very, very long time. Okay? For large proteins, this would be incredibly slow to do. And it is quite robust, though. You literally take an amino acid, cleave it off, figure out what it is. And you just keep doing that until you run through the entire amino acid, right? Yeah, very robust, very slow. All right, and so this is actually an Edman degradation that you're seeing sort of in a diagram here. Again, we're going to label this on the end. Right there's your labeling. Okay. We then cut off the first amino acid. We're then going to take that and we're going to figure out what it is through chromatography. Okay? And then we're going to cut off, label the next amino acid 2, cut it off, figure out what it is through chromatography. Okay? And we'll just keep doing that over and over and over each time we have shortened our peptides. Okay? All right. So the peptide sequencing is now more commonly performed by tandem mass spectrometry. It's a far faster technique and more precise and accurate typically as well. So it's just a better technique as long as you have this set up properly. Okay? If you don't know what the fuck you're doing and screw this up, you'll not know what you're doing and you'll get wrong results, right? Which I guess you can say about anything. The tandem mass spectroscopy, so your precursor ions are going to be broken into smaller chains, and the product ions can then be passed through a second mass analyzer. This is the tandem part, by the way. Tandem meaning literally one mass spectroscopy followed by another. Okay? Okay. And so what we can see here is we actually have, if we look very closely, each time we break off something, we're going to have a mass to charge ratio that's going to correspond with it, right? And as we break off less, we'll have an increasing mass to charge ratio that corresponds with each amino acid. So see that we're again breaking them off sequentially. Or we don't even have to necessarily break them off sequentially, but we know that if two things have a number in between, if you have something that has a number in between two different things, it's the one that you had one less amino acid, right? Okay, so it doesn't actually matter the order here that this comes out. What you'll see here is that literally each of these peaks here will correspond to losing one amino acid, right? Yeah. Okay. And so you get peaks. Each one will be a different amino acid loss. Each amino acid has a slightly different molecular weight, doesn't it? And so you can tell which one has come off. Pretty nice, right? Okay, so the Edman degradation is going to be limited to peptides of 50 residues. Not all the peptides in the reaction mixture are going to release amino acid derivatives at each step. So you can actually get skips, and it is limited to 50. Sequences of long peptides are going to be done through mass spectrometry. This will yield complex spectrums that are difficult for people to interpret. To sequence an entire protein, the protein is going to have to be chemically or enzymatically cleaved to yield peptides of fewer than 50 amino acids, and then typically you have to sequence them individually. The peptides will then be ordered by performing different cleavage procedure to generate overlapping peptides, and then you can go, okay, this one has 20 amino acids that are the same as this fragment, they match, right? And then it has 30 that are the same as this fragment, those match. And so these must join here like this, okay? And most of this is actually done by programs now, to be honest. The software is quite robust for this, so it's actually, a lot of people do it and they don't even know what the fuck they're doing. They're like, magic machine, tell me. But anyways, these are the specific cleavages of different polypeptides, and this is how you can then cleave your peptide into multiple different fragments that you could then run, okay? Most of these are the enzymatic cleavages. These are the enzymes that can do cleavages that we found and purified and you can buy, okay? These ones are the chemicals we've found that can do cleavages, and you can run these chemicals and have a very known reaction, okay? By the way, if you look very closely, you will notice that these chemical cleavages are just straight up the organic reactions we've talked about previously. Okay? Like, that's what that is. It's just things that, you remember all those chemical reactions we talked about that tend to cause cleavages? Yeah. There we go. Okay. So here we have the cyanobromide. This is where it's going to cleave. It'll cleave on the carboxyl side of methionine residues. So you'd find any methionine within your polypeptide. On the carboxyl side, everybody knows what the carboxyl side is, right? It's the side that would have the oxygen, the carbon and oxygen. Okay. Not the side that would have the amine part of the amide linkage, right? It'll cleave and leave you with a fragment starting with that amine that used to be in the amide, right? And your new amino acid, the one that's next to the methionine. Okay, so anyways, these are the different cleavage sites for these different reagents here. Does anybody have any questions about that? So this would actually be what we're talking about, the overlapping peptides. So here we've actually digested this peptide here and then sequenced it, okay? And so we've used trypsin and chymotrypsin. Notice the trypsin and chymotrypsin down here. Okay? Okay. And so what we've done is then we can take and arrange these fragments so that we have some sort of overlap, right? So we arrange the fragments, and what we can see here is if we look at the blue one, this right here is this fragment right here, right? And because of these overlaps with these red fragments, notice the Ella, TRP. Okay, that has to be right there, doesn't it? And we know from the directionality of this that this other one has to be in front of it. Okay? And see how we've done that now? Quite nice, right? That's that same puzzle solving that you're getting to kind of know more about in organic 2, and we had started talking about in organic 1, right? Okay. All right, so the disulfide bonds are going to be cleaved by reduction with thiols, such as B-mercaptoethanol or dithiothreodol. The disulfide bond reformation is going to be prevented by the alkylation of your cysteine residues with iodoacetate. Okay, and so this is actually the alkylation right here. And this is what's going to cap those thiols so they don't just rejoin. Okay? Okay, so even with today's techniques, determining the amino acid sequence of a proteins actually can be very challenging. Keep in mind we have to cut these things up into fragments, right, to do this and get overlapping to figure out where these things are, even using the MS techniques, the mass spectroscopy techniques. And so when you're talking about a 10,000 amino acid protein, that can be a lot of work, right? Okay. The software is pretty good, and it actually can do a lot of expansion of humans, give us like possible matches, and then we can do some more spectroscopy work to eliminate those. Okay, but just by single technique, this one single technique, you may run into proteins you can't just do automatically. Okay, you could have multiple different possibilities that would all work, or it could take a very long time to figure out which one. So for sequencing very large proteins, a complementary experimental approach is the best method. This is going to be based on things like recombinant DNA technology, which will be more efficient. So stretches of your DNA can then be cloned and sequenced. The nucleotide sequence can then be translated. This will reveal the amino acid sequence that encodes the gene, encoded by the gene, that encodes the protein. The amino acid sequences of proteins, these can be compared to identify similarities. Remember that the amino acid sequence will ultimately give you the protein structure, and function and history. But it can be difficult to tell what exactly that would fold into if it hasn't been seen before. Okay? Because again, all the secondary structure is derived from the primary structure, right? We had talked about that there's certain amino acids more likely to cause certain secondary structures, and so on down the chain. But again, once you get thousands of these, it can be very difficult, right? All right, so this is the comparison of the sequences of the same protein from different species. Is also going to yield information about evolutionary pathways. The amino acid sequence searches can reveal the presence of internal repeats. Sequencing information can identify signals that determine the location of the protein or processing signals. Sequencing information can also be used to generate antibodies for the protein, because you know if it has these groups on it, you need to match those groups. The amino acid sequence can be used to generate DNA probes for specific gene encoding proteins. You guys have maybe even heard of this. They have like these new cancer tests that you can send in your DNA sequences, and they can look for cancer markers. And so you don't need to do screenings for those cancers as often. Okay? Because some cancers are very, very dependent upon having the genetic encoding for it. This is the repeating motifs in a protein chain. This would be a very good motif to, say, target with an antibody, right? If you have repeating motifs on it, because it would have a huge interaction with antibodies. Individual proteins can be identified by mass spectroscopy. The peptide mass fingerprinting is actually very similar to, remember how we talked about infrared spectroscopy and there's the fingerprint region? Okay, so yes, you could resolve each individual band in one of these mass spectroscopies, right? But it takes a lot of work. However, what if you just want to tell if that's the same protein or not? Do you actually have to tell what every single band is? No, you just need a known sample, run it, and you'll get a fingerprint, right? You then compare it to new ones. If they look the same, it's the same thing. There you go. I know. This will involve a very specific regiment of protein cleavage, chromatographic separations of the resulting peptides, and then mass spectroscopy. And you just run it the same way every time, and you'll get the same fingerprinting, basically, assuming no evolution, every time, right? This right here is going to be proteomic analysis by mass spectroscopy. And as you can see, here we have different spectra. And you can see how complicated those look, right? Okay. But you can see that you get different peaks for different variants, okay? Okay, so a proteome. This is going to be an entire set of proteins expressed and modified by a cell under a particular set of biochemical conditions. So unlike the genome, the proteome is not a fixed characteristic of a cell. The genome is. The proteome, the stuff that's expressed, will be changed depending on your environment, right? And a lot of factors. It can be the amount of stress it's under can change the proteome, okay? Synthetic peptides can serve as antigens, produce antibodies. We insert a synthetic peptide, and so now we can have an antigen that will produce antibodies for that peptide. We can be used to isolate receptors, so synthetic peptides containing, for example, formal methionine, FMET, you may have seen that before, FMET, have been used to identify receptors on white blood cells. And this is FMET, by the way, in case you're wondering. It's the methylated thiol group here, okay? They can serve as drugs, of course. They can bind into a region and then inhibit any activity of that enzyme, right? Or they can then be used into the structure, and now you're in a methyl group where you should have a thiol, right? Obviously, that's no longer going to function as an enzyme, is it? Yeah. This is a lot of the antivirals, by the way. You basically feed the person a source of amino acids that the HIV uses a lot and don't function in the way they should. And so the HIV then uses them and makes non-functioning HIVs, right? Or I guess it'd be AIDS at that point usually, but. Help define the rules governing protein folding. We can put in different various groups here that have slightly different charges and sizes and different hydrophobic versus hydrophilic properties. We change that side chain and all of a sudden it's going to change how that folding can happen, right? And so then we can test. This folding happens because of thiol bridge. Can't make thiol bridge anymore. Does it happen? Okay. Okay, so this is vasopressin, and we have a more synthetic, more stable synthetic vasopressin analog. Okay, so right here we can see the analog right here. So this is the antidiuretic hormone, ADH, right here. And here we have a more stable variant. This is desmopressin. Patent. Don't try to buy my patent, damn it. And you can see there's just a slightly different orientation here. And we went ahead and just cut off the the charged amine group up there. And that actually makes it more stable. And so you get longer term, more effect from it, right? All right, so solid phase method. Notice, by the way, by switching this site here, what have we done? We're now the other isomer, aren't we? Yeah, we've swapped right to left hand, haven't we? Okay, so the synthetic peptides are going to be synthesized in a stepwise fashion. This is for solid phase method, with carboxyl terminus of the growing chain attached to an inert matrix. This is what gives it its name, solid phase. Literally, we have attached it to a solid phase, and we're now growing it off of that solid phase. So then we can collect them, right? So some of the groups must be blocked and others must be activated to prevent unwanted reactions. Keep in mind, by attaching them to a solid phase, we're actually doing most of the stuff that you would associate with a catalyst activity, right? Much like the catalyst that we use for hydrogenation, right? We're aligning them next to each other so they bang into each other a lot, okay? So you really have to watch out for that. The role of protecting groups in solid phase method, this is one of the protecting groups you're going to be seeing here. So here we have TBOC amino acid, and we have an FOMC amino acid. These protecting groups, right here, this is one. See how it's bulky and protecting that end chain? And there we have an even more bulky group, right? Protecting that end chain. Okay. Those are going to keep these from reacting when we don't want them to react. Okay? There are also groups, protecting groups that you can detach when you want the reaction to occur, right? You just have to do a reaction to get them to detach. And then you're ready for the next one. Okay, so the protecting groups in this case is removed by trifluoroacetic acid. This will free the amino group. We can then proceed with the next coupling reaction once that's freed. Okay. All right. So DCC, this is going to react with carboxyl group of the incoming amino acid. This will activate the amino acid for the peptide bonding forming reaction. Remember, in these cases, we're synthetically building these things usually onto a scaffold, essentially, right? And so we need to activate these. Normally, that activation would have been done by the enzyme sets in the body, right? Okay, so here we have that solid phase peptide synthesis actually happening in a stepwise reaction. Here we have the activated resin here. And you can see it's resin. It has this activated chloride group here. By the way, notice chloride good leaving group. Here we have that oxygen group, which can attack, right? Straight up, like an SN2 style reaction, right? Okay. I know. The damn organic chemistry. So again, the first thing we need to do is we need to anchor it. So we anchor it onto the resin, and this is our anchor right here. Then the next thing we're going to do is we protected this amine here so that we didn't get reactions we didn't want. And so now we're going to take off that protection group, the TBOC in this case, with that trifluoroacetic acid, and that's trifluoroacetic acid there. Now we have an open amine. We have an activated amino acid right here, activated with DCC. This is then going to come along. It's going to react, form the amide linkage, and we've linked on another amino acid. Notice that we keep protecting those protecting groups. What would happen if we did not protect this amine here? This is a very reactive, going to react with amines, right? So if two of these were to come together, two of these blue ones, what would happen if we didn't have a protecting group on one? Yeah, and we get these little fragments floating around, right? That could then bind to stuff that we don't want to do. Okay? And so that's why that cycle of protection, taking off protection, happens. Right? So we can prevent that. Does anybody have any questions about this so far? Yeah, we basically just do that stepwise until we get the sequence of amino acids we want on this. Okay, so the native chemical ligation is going to link two synthetic peptides. Here we have the trans thioesterification. So here we have peptide fragment number 1. We have a peptide fragment that we have a thiol group attached to. And then we have a peptide fragment number 1 that has a sulfur attached to it, that carbonyl. Remember that sulfur is a pretty decent leaving group, right? And so we had seen similar reactions back when we talked about sulfur towards the end of the organic chemistry. And so this is that trans thioesterification. Notice you have a thiol reaction. And esterification is this part right here. See that? Okay. Okay, so what's going to happen is we're going to link this sulfur here onto the carbonyl. That is still quite a reactive group. So what's going to then happen is the amine here is actually going to end up having a rearrangement. And so we then will form an amide linkage here, and we can take that thiol off if we need, or we can do other reactions with it. Okay? Okay, so X-ray crystallography. This is going to reveal the three-dimensional structure in atomic detail. This is actually how the majority of three-dimensional structures for proteins have been found as of right now. Okay? The underlying physics principles of X-ray crystallography, some of the things that you need to keep in mind, are the electrons of the atoms are going to scatter X-rays. These scattered waves are going to recombine. The way in which the scattered X-rays recombine is going to depend only on the atomic arrangement within. Okay? Now, does anybody notice something about this word, X-ray crystallography? What word is within that? Crystal. And so is this a free-floating enzyme, protein, whatever? No, this is the crystallized form, right? Does that matter? Yeah. There's a different structure for a crystal than there is free-floating, right? How much of a difference? Depends, okay? But we hope not so much of a difference that matters, and we get nothing, okay? This is actually the X-ray crystallography experiment going on. We have here an X-ray source. We shoot that X-ray source into a crystal. In this case, we have a protein crystal, but you can do this for all sorts of things, by the way, and get three-dimensional structures for them. And what's going to happen is when the X-rays hit that protein crystal, they're going to hit the electrons and the proton and get scattered from those. Specifically, the protons are going to be the big ones, right? But the electrons are also going to cause scattering of those X-rays. Now, where is it going to scatter the most? Where you have the most density of electrons, right? And so how it scatters and hits the detector, by the way, they used to literally use a piece of film and then wrap it around. And then the X-rays would hit the film and they'd tell where the scattering dots are. Okay? Now they have a digital detector and it does most of the work for them. And so the next part is going to be, we take that diffraction pattern, we interpret that diffraction pattern, and this is going to give you a density map on how it's scattered. Okay? And so this is what an X-ray diffraction pattern looks like, by the way. Can you see the protein? No, you can't. You need some pretty thorough math to do that. This is actually the interpretation of the electron density pattern. And you can kind of tell what the protein looks like from that, don't you? Now, if we start assembling the amino acids within this, this is what it would look like for the backbone. Okay? Now, this is not something we actually get from experimental data itself. Okay? This is just matching the experimental data we got to what we think it looks like. So the resolution will affect the quality of the electron density map. Depending on what sort of resolution you have, you're going to get a much looser or much tighter electron map. For example, this is a 1.04 angstrom, and you can see that we can actually resolve like little gaps between in the middle of molecules and stuff. Okay? But when we have a larger number, for example, up at 2.75, we can no longer resolve that, right? Basically, past 1.75, we lose that resolution. Okay? We get up to 6, and we can't even resolve, like, what the hell's in there, right? Everybody see that okay? Okay, so your resolution makes a big difference on what exactly you can resolve, but you can get overall three-dimensional structures. Note, however, low resolution can cause you a lot of problems. For example, this looks like it comes back together up here, doesn't it? See that? Okay, and it can really trick people. By the way, this is one of the original pieces of data that was determined the structure of like DNA. X-ray crystallography has been around for a while. So the nuclear magnetic resonance, this is the NMR spectrum. If you use it in a hospital where with people that don't really know what's going on, it's called MRI. But the software is really good, so they don't really need to know much of what's going on. Based on the fact that these certain atomic nuclei are going to be intrinsically magnetic and can exist in two different spin states when an external field is applied, the nuclei of the sample absorb electromagnetic radiation in different frequencies. This is termed chemical shifts. And you guys are currently learning this right now, if I'm not mistaken. Okay? In organic too. The chemical shifts are going to depend on the environment of the nuclei, and the environment depends on the protein structure. To be honest, we're not actually going to, this is basically all the NMR you're going to get in this class. Okay? The biologically important nuclei that you would monitor are these ones right here. You can monitor any one that's not even. Okay, that's uneven. That doesn't mean even in number, like here's nitrogen 14, right? That means that it has one point that's irregular on it, and so the spin field then is not symmetric. Okay? Kind of like a top. If it's not even on every side for the weight, it doesn't perfectly spin, right? Same thing here. You do have to have something that occurs at a reasonable rate in nature, or else you're going to have such a weak spectra that you can't actually see it. Okay? Anyways, these are the important ones. This is the natural abundance. The ones that have less natural abundance will give you much weaker spectra, which can be problematic. Okay? But anyways, this is why, for example, carbon's not used a whole lot. Carbon-13, when you're looking at biological samples, but can be used. It's just more difficult. You need a very sensitive instrument. So the basis of NMR spectroscopy is this right here. As we increase the magnetic strength, what happens is the difference in the spin states of the nuclei, i.e. which direction it's spinning, counterclockwise or clockwise, right? Or up and down being the preferred terminology, okay? Is going to increasingly separate in terms of energy. So if you have a very, very large energy field, you can separate the energies between them quite a bit, okay? And then what you're going to do is you'll radiate them. The lower energy population will flip into the higher energy population. And that gap in energy right here corresponds to the energy of this irradiation, okay? And so what ends up happening is you get a transition between these spin states. When they come back down to the lower energy spin state, that's going to re-emit energy, right? And that energy will be a very consistent electromagnetic frequency, okay? And that will be corresponding to this energy gap. And this actually tells us how much of an energy gap it takes to do that, okay? Which is what we're seeing in NMR. And so you can change your magnetic field to have different things split at different energies, which then match the radio frequencies you're pulsing in. How they come back down to the lower energy tells you what that energy amount was. So this is one-dimensional NMR spectra. This is the most common stuff you see. And so the resonance spectrum is going to be obtained by keeping the magnetic field constant, and you're going to vary the frequency of your electromagnetic radiation. Typically, this instrument is going to have a 300 megahertz or 3,000 megahertz magnetic field. Okay? And that's what you guys have probably been looking at, is like 300, 600s. And that's the strength of the magnet, right? And you're looking at one-dimensional NMR. Okay? And you probably recognize this right here, don't you? This is an idealized spectra and the stuff we usually give you. This is a real spectra right here. Not as clean. Real world, right? Yeah. Does anybody have any questions about this? Recognize the reference TMSO giving you the 0 point, right? This is the downfield shifts for each different group on here. Okay, notice this is a hydrogen one. All right, so two-dimensional NMRs where you guys are not going to see this probably in organic at all. Probably not even going to touch on or talk about. It is the thing in biochemistry, okay? You can resolve a lot more details. And so in two-dimensional NMR, the spin in one nucleus is going to be altered, and the effect of the spin on the neighboring nucleus is going to then be examined. So for example, the nuclear Overhaus enhancement spectroscopy, NOESY, is based on your nuclear Overhaus effect This is going to be an interaction between nuclei that are proportional to the distance between them. And so what you're going to see here is we're going to have two different NMRs we've taken, okay? And in this case, we're not shifting, we're not worrying about intensity. Basically, the intensity would be a third dimension, but we're not going to talk about that, okay? And so we take two of these spectra, and we plot one's chemical shift in one axis, and the chemical shift for another one in another axis, okay? And how they correspond here, these chemical shifts here, is going to tell us how far away those two groups are from each other, because they will affect each other's chemical shift depending on their proximity. You've actually seen this to some degree in this right here. If they're very close together, they affect each other's chemical shift by that little tiny amount, right? We're just looking at it by using two different ones. So we're getting a greater effect from it. Okay? Ta-da! Isn't that cool? Yeah, and so what that does for you is, number one, you can tell where the hydrogen is based on its chemical shift. Is it near carbonyl? Is it near nitrogen, right? And then secondly, you can tell how close are those hydrogens together. You can also do this for things like carbons, nitrogens, oxygens. You could do this for irons, by the way, for like hemoglobin, right? Yeah. And so you can actually do this for a lot of different groups. Utilizing this, we can actually figure out the three-dimensional structure of a polypeptide. Okay? It's just we have thousands of these damn things to figure out. So it can take some work, right? But there's really good software that reduces people's burden and helps out doing it. Okay? This right here is actually what it looks like in real life. Like, again, this is an idealized version, right? And those splits that you guys have talked about, that's that ring structure. See how there's the rings? Okay. This is what it looks like in real life. It's kind of hard to see because we're not very zoomed in. But if you look very closely, you can see they have like densities to them, right? They have like coloring, where it's more concentrated, less concentrated, more concentrated on these dots, okay? And so that's actually telling you those splits that we had talked about before, that ring structure, okay? That you guys talked about in Organic 2, excuse me. I usually teach the Organic 2 along with this, so Evans talked to you about before. So anyways, these structures are going to be calculated on the basis of the NMR constraints, and so you would then calculate the structures based on how they're showing up on that spectra, okay? And that will give you, using a fairly standard calculation that you can do, like usually you'd use a computer that does it all for you, would show you how these match up. For example, these hydrogens here are close together. That means this has to fold in such a way that those come in proximity, okay? Okay, the family of structures derived from NMR experiments, we can see that right here. This is what the structure would look like just based on the NMR experiment. Notice at the ends here where they don't fold together. See how it looks like frayed rope out here? That's because there's many possibilities for those, right? Because the only places we can see with this are where it folds back together. Okay? We can't see the ends where they don't come in contact with anything else. All right? Okay, so the cryo-electron microscopy, cryo-EM, EM being electron microscopy if you ever wonder, is a thin layer of protein solution. It's going to be prepared in a fine grid. It's quickly then frozen to keep it from shifting a lot. You have to take the temperature down really fast, okay? And that keeps it from doing things like crystallizing and stuff, right? It just takes it down so it can't move anywhere. This is going to trap your molecules in an assembly of orientations. The transmission electron microscope is going to be under vacuum conditions, then going to be used to obtain the two-dimensional projections of that structure. The computer is then going to take that series of two-dimensional structures as you rotate these different directions. You'll have different proteins that are in different directions in your sample. And you're then going to assemble those into a three-dimensional structure of the outside. This is actually a lot of what like the structures we see in our book are based on. Okay? And you can kind of see them right here. See, this is what you actually get right here. And then you can abstract that using a ribbon abstraction here. And that's what we see in our books, right? Okay. Notice these are the different sides to it. Can you see that? We just rotated it in different directions. We're literally going through and just looking at different ones. Or we can rotate our crystal if we need. Okay? Wow, I actually finished this, didn't I? Holy shit. 1 minute over, but okay. Did anything I just said make any sense? Yeah, okay. Let me know if it didn't, and I'll go back and cover regions, okay? In a separate video at another time. Please talk to me about homeworks at all. You have a wonderful day. I'll see you Wednesday.